Over to you, Lonnie.
Excellent. Thank you very much, Shane. So thankfully the Diplomix team have given a really good introduction to a lot of the workflows. So maybe I can make up some time in going through this. So as Shane said, my name is Lonnie van Zyl, and I'm from the Institute for Microbial Biotechnology and Metagenomics. So we're a research institute based in the Department of Biotechnology at University of the Western Cape. So let's get right into it. So our story starts back in 2007. The institute was founded by Professor Don Cowan. So for those in the microbial space, you'll know that Don is a prolific microbial researcher in South Africa. And his special interests are extremophiles. So he went all over the world collecting a variety of samples. And of course, the group isolated a range of microorganisms. And since Don's departure from the institute, we've carried on the tradition of building out our collection of microorganisms, not necessarily extremophiles, but mostly from marine and terrestrial sources, to the point where in 2021, we had amassed quite a range of bacteria from a variety of different sample sites. And through the lobbying work of Dr. Anita Berger, who is in the audience, so she's our research and innovation manager, we managed to secure one of the biodiversity biobanks of South Africa to formalize this microbial collection into a biobank. So as Tim mentioned earlier, BBSA, or I think Shane did, is one of these South African research infrastructure roadmap initiatives. So when the idea was proposed for 1KSA, it was only natural that the two infrastructures come together to work on sequencing a range of microbial genomes. All right, so our commitment to the project was to sequence 500 bacteria from this collection. And that's a target that we've now met and surpassed. So today I'm going to tell you a little bit about the lessons learned along the way and how having this sequence information might be of use going forward. All right, so it turns out sequencing the microbes that you've got in your collection is definitely the way to go. So the American Type Culture Collection and the DSMZ are the two largest microbial collections in the world, and they've started doing this for their collections. So it's good to follow suit. And as Shane had mentioned earlier, there's sort of two main things that you could probably do if you have the sequence data in hand. One of them is biodiscovery. So you can look for a gene or genes of interest, and you can valorize that in any way that you like. The other thing you could do is you might have some sort of ecological question in mind. So you might be interested to look at the diversity of microbes associated with a particular environmental niche or a particular host. You may want to do some genomic comparisons to see how these organisms evolve both in time and in geographic distance. Or you may want to do functional studies where you use the genome information to sort of recreate the metabolic pathways that these organisms have to look at both the interaction between the microbes, but also with the environment if you're looking at something like nutrient cycling. So I've colored these blocks, green and orange. So the green blocks is where I think normally you would want pristine, complete genomes. And in terms of diversity studies, I think we can still get away with suboptimal assemblies. Of course, we always want perfect genomes, but that's not always the reality. So the other thing that is very useful in having the sequence information is to de-replicate strains in your collection to make sure that you've got representatives from the myriad of genome types that you can find. And the other thing that I find really important is with all of this sort of culturing and DNA isolation and sequencing and analysis going on, this is a fantastic opportunity to train technicians and interns and students and to build local capacity in this regard. All right, so I quickly just want to take you through the 4 collections that we selected microbes from to sequence for the project. The first one is a collection that we call Pharmacy. So Pharmacy was a 4-year program, EU funded, and the idea was to isolate microorganisms from marine invertebrates and then screen them for compounds of interest, notably anti-cancer, anti-inflammatory, antimicrobial, and neuroprotective activities. So that was spearheaded by Prof. Marla Trindad, who's also the director of the Institute. So we selected just over 330 isolates from this collection. We did isolate about 3,000 during the project. The next collection are a range of actinobacteria, again from marine invertebrates and marine sediment. And this was contributed by Prof. Marlies Hill at CPUT. And we elected to sequence just over 120 from this collection. Then we sequenced 60 genomes from Dr. Brian Kirby's collection. So again, these are actinobacteria, but this time from rooibos and aloe rhizosphere. So her and her students have, over the last couple of years, really focused on plant microbe interactions. And these are certainly the most photogenic of the bugs that we sequenced. And then lastly, we sequenced about 40 soil isolates from these termite mounds. So here, very colorful, you can see them with the wildflowers growing on top of each of the termite mounds. And I'll say something about that a little bit later. All right, so you've now seen several workflows, and ours is no different, really. So we start off with the microbe that we culture in a broth, and we then use a commercial kit to bust open the cell and extract the DNA. So this is actually one of the little lessons learned along the way. We initially started out with a different kit, different manufacturer, but we switched over to the Zymo kit because we find it does allow us to access some of the more difficult to break open bugs. Once we've got our DNA, and so maybe I can take a short pause just to say that microbes are a lot easier to work with than the plant genomes that you just heard about, and also some of the macrofauna. So even though that is the case, it has challenges all its own. But we find it comparatively easy to extract clean high molecular weight DNA from these bacteria. Once we've got that, of course, we can make our sequencing library and actually generate the sequence. And now you've heard this several times. The data then gets transferred to DERESA for long-term storage and to CHPC to do some of the data pre-processing, assembly of the genomes, and some of the analysis. The genomes in general and the data sets are small enough, rather, that we can actually do a lot of it in-house as well if we need to. All right, so when you do an exercise like this, I ask myself the question, so what can you really learn from this? And for me, I've broken it up into these two sort of themes. The one is all about the technical aspects. So how do you go about isolating high molecular weight DNA? How do you make the best library? This sort of thing. And then secondly, what can you learn from actually studying the genomes themselves? So the biodiscovery and ecology themes that I mentioned earlier. So if we start with the technical side, how do we go about sequencing these genomes best? As of today, we've sequenced 547 isolates. And so here I just show the breakdown of the number of genomes that fall into these bins. And the bins are genomes that have assembled to between 1 and 5 contigs. So as you've heard earlier from the true bioinformaticians, I'm a weekend bioinformatician. One of the things that we look at, if we look at the quality of a genome, whether we've managed to recapture all of the genetic information, is to check for these conserved genes. So in the microbial space, we use a different tool called CheckM. It essentially does the same job as BASCO. And so if we use that as a metric for saying what number of high-quality genomes we have, then we've got 410, so about 75% of these genomes are of really high quality. We also add another metric to make it a little bit more stringent, and that is to look at the genomes that assemble between 1 and 5 contigs. And where this comes from is if you look at the GenBank database, there are about 250,000 bacterial genomes on there that are scored as complete, and the average contig size for those is 5. So in case, if you remember back, if you're not a microbiologist and you tend to think of bacteria, you tend to think they've got a single chromosome and maybe 1 or 2 plasmids kicking around. But that paradigm is changing rapidly as we're using long-read sequencing. So as it turns out, there are very many bacteria that have multiple chromosomes. So other nuanced things that happen when you're doing this, as you're culturing the microbe, very often some of the prophages that are stuck on the chromosome will also decide to go lytic and pop out, and you sequence them along with the genome, and they end up as a contig alongside the genome. And so if we do that, then we've got 307 genomes so I would score these as remarkably high quality. So the question then is, if you take 547 and you subtract 307, there are some genomes that didn't quite make the grade. So what do we think about those? And that just comes down to genome coverage, or the amount of data that you manage to get for those particular genomes. So the histogram and the violin plot here just show the coverage distribution for our various genomes that we managed to sequence. So the average coverage was 104, which doesn't look too bad. And Oxford Nanopore actually recommend for de novo bacterial genome sequencing and assembly that you cover the genome more than 100 times. But if we look at the median, then we see that's 57. So that means that half of our genomes are sequenced more than 57 times, and the rest less. If we look at the quartiles, then we see 75% of genomes were covered more than 16 times, but the mode is 4. So that doesn't look particularly good. But I hope that I can convince you that even though some of these genomes were only covered 4 times, that they are not useless. They are still, in fact, quite useful. So put another way, we generated a total of 490 odd gigabase of sequence, which for those genomes, assuming a genome size of 5 megabase, should have been enough to cover each one of them 180-fold. So here I try and make excuses by showing that even though this selection of genomes that I show here were only covered 4 times, we actually managed to reconstruct the genomes in a fairly small number of contexts. So if you're not in the NGS space, these numbers won't really mean much to you. But for example, when we used to sequence with Illumina MiSeq, where the read length is 300 base pairs, the best ever de novo assembly that we managed to get was 30 contexts for a bacterial genome. And so if you told somebody 10 years ago that you could cover a genome 4 times and get this number of contexts, they would tell you you're lying. So this really isn't all that bad. Also, if you look at the N50 score for the assemblies, it's really not that bad. And just to drive the point home, below here, I show the comparison of 2 secondary metabolite pathways from 2 of these genomes compared to the related genomes on GenBank. And so, yes, there are problems here, but it was actually good enough for a tool called anti-smash that we use to identify these secondary metabolite pathways to, first of all, identify the pathway. And of course, knowing where these errors are, that is absolutely fine. These genomes are perfectly fine for a range of questions that you may want to ask. And particularly for diversity studies, we think that they should be absolutely fine. All right. So in a nutshell, what were the reasons for the poor coverage and how do we improve on this going forward? So my number one reason here, I say, is the type of bacteria. And what I mean by that is that we believe that the genomes are chemically modified. So it is well described in literature. Our genomes do the same. But I don't believe that the range of chemical modifications has been fully explored. And this has 2 consequences. The one is when it comes to creating your sequencing library. So we opted to use the rapid barcoding kit, and that is a library prep technique that's based on a transposon that can integrate and cut the DNA. So if the DNA is chemically modified in such a way that the transposon can't actually recognize the DNA as a template, you literally won't create a library, or you create a pretty poor library. And second of all, the way ONT sequencing works is that your base caller has to have been trained to recognize these modifications on the DNA in order for you to convert the squiggle file to a base call. And if it hasn't been trained on that, you can generate all the sequence you like. You literally can't interpret the data. So the way around this is to perform whole genome amplification on all the genomes going forward. So that is going to be our solution. Essentially, this replaces all the modified nucleotides with canonical DTPs, and you essentially end up with a perfect template for sequencing. So for now, we are going to stick to the rapid barcoding kit. Oxford Nanopore tell you, this is well known, that if you use these rapid barcoding kits, you do get lower output. But there are 3 reasons why we're going to stick to this. We've done several tests. So once we do whole genome amplification, we get more than enough output using these kits. The second is literally the convenience of using these kits for creating libraries. And the other is the cost. So if you do a ligation sequencing library prep, you've got to buy another kit from NEB, and this raises the price. And really, I come back to the convenience. These kits are fantastic if you're trying to train people to do NGS. then this is an absolute breeze. So the last point here is the platform itself and the nanopore software. And this is really where I think nanopore can focus a little bit more and improve their offering, because a lot of the early sequencing was done on the Mark 1Cs, and we would often find them stuck. So whether the machine itself was overheating or it's actually a software glitch. So between the two, we would lose a lot of sequencing capacity, especially in that golden 24 hours. And so what we found is that the Promethean platform is actually a lot better. So going forward, especially if you're doing large runs, we'll try and do that on the Promethean platform. Shane, if I do run long, yeah.
Go 5 minutes.
Oh, snap, okay. All right, so the other thing we could do, as I alluded earlier, is we can study the genomes themselves and see what we can learn. So here I've just sort of broken down the genomes that we sequenced into the different phyla. So in the end, it was composed of 105 genera with 1 potentially novel genus and 129 potentially novel species. So the asterisks are there because it is not currently accepted to classify an organism as a new species based on genomic content alone. So then I wanted to just give you an example, 2 examples from our own group when it comes to asking an ecological question or asking a biodiscovery question using this kind of information. So one of these studies that we're involved in at the moment is investigating carbon sequestration in termite mounds. So as you travel up the road, the N7 going towards Namibia, if you look out the window on the side of the road, you might see these low relief little hills. And if you didn't know, or somebody didn't tell you, these are actually termite mounds. So they're not the classic, spiky termite mounds that are quite characteristic elsewhere in Africa. But as it turns out, people have found that they seem to be significant carbon sinks. And so here you can see a slice through one of these termite mounds, and all of this white deposited material is calcium carbonate. And it's thought to happen through a process called microbial-induced calcite precipitation. I'm not an expert in this. So the main way it's thought that these mounds serve as a carbon sink is, of course, the plants photosynthesize, capturing CO2 from the air. The termites then obviously feed on the plant material and take it down into the soil and deposit it in 2 ways, both as physical pieces of the plant, but also as frass. So that's pictured up there, termite droppings. So through the photosynthetic process, the plant produces a lot of calcium oxalate in the plant tissue. And you find then that the frass, as well as the plant material that they take down into the mound, is obviously rich in this carbon source. So this work was initiated by Kathy Clark and Michelle Francis from the Soil Science Department at Stellenbosch University. And they did a hell of a lot of work to try and understand the chemistry that's going on here. And they've got a pretty good grasp of it, but they eventually found that there's a missing puzzle piece. And it turns out it's likely the microbiology. So what are the bugs doing to move all the carbon around? So that's how we became involved in the project. And this is work done by Ryan Bird, one of our master students. So he took some of that frass material and made growth media where that's the only carbon source, and managed to isolate a range of bacteria on there. And then through the 1KSA program, we managed to sequence the genomes. And so what Ryan's done is tried to identify the enzymes that are involved in this process. And the next job, so this is still an ongoing study, is to clone some of these out and actually characterize the enzymes and look at the biochemistry of this for us to have a better understanding of the role that the microbes play in this carbon sequestration process. All right, so that's ecology, then biodiscovery. So one of the main themes in the Institute is finding compounds or enzymes of interest that we can use in some way. So one of the classes of compounds that we're particularly interested in are lantipeptides. I'm not going to explain all of that, just to say that these are ribosomally synthesized peptides. So they've got a leader region, a core region, and what makes the lantipeptides unique is the installation of these lanthionine rings. So these are thiether bridges, and It happens in a two-step process. First, dehydration of serine and threonine residues, followed by cyclization to install the bond. A lot has been discovered or described about these lantipeptide classes from gram-positive bacteria, but virtually nothing is known in gram-negative bacteria. But it's not because the pathways aren't there. Now that people have sequenced so many bacterial genomes, we actually see the chock-a-block full of these things. But people haven't picked them up because in gram-positive bacteria, these often display antimicrobial activity. So our thinking is that either they don't have antimicrobial activity from gram-negatives, or that they're not expressed under lab conditions that people tend to culture these organisms. So we have identified this as an understudied class. And so this is just one example. So several years ago, one of our PhD students, Ross Vermeulen, identified one of these class 1 lantipeptides from a marine bacterium called Thalassomonas viridans. He looked at the chemistry to see where these lanthionine rings are installed, and he looked at some of the stereoisomers that are formed. He did the usual thing, looked for antimicrobial activity and didn't find anything, and in desperation eventually tested this in a zebrafish model. And this is actually one of the first demonstrations of a gram-negative lantipeptide having some sort of bioactivity. So this is not necessarily the activity of this compound in its host organism, but he found that one of the peptides had a stimulatory effect on the zebrafish and another one had a sedative effect. And if you combine the two peptides, they nullify the activity. So yeah, a lot of detail I'm glossing over. All right, so again, we went mining in genomes that we sequenced during the 1KSA program in bacteria that come from Kalk Bay. So we've identified a whole bunch of these class 1 and class 2 lantipeptides, and this is all part of another 4-year EU-funded program called Blue Remediomics to try and valorize marine resources, sequences, or microbial collections. And so these are in different stages of being characterized in our lab, and of course we will be screening these for bioactivities, and hopefully we find something. All right, then lastly, just to say, echo what Shane has said, and others, that all the protocols that we used for sequencing the bacteria are on the Zenodo page. If you're interested in working with these strains or with the genomes, please make contact with us. This is meant to be a national resource. We obviously have a first look at the genomes, which is great for us, but we certainly want to farm this out so that other people can work on it. And we've already had requests from Rhodes University as well as Free State, and we've shared some strains and genomes with those folks. All right, I just have to say a really big thank you to Tim and Shane and the rest of the Diplomics crowd for partnering with us. This has been a very fruitful relationship, and long may it continue. So we're really grateful for it. There are a lot of other funders that we have to thank, in particular the Biodiversity Biobanks of South Africa for funding the biobank, as well as giving us money for Stephanie back there, who's the biobank manager. And then lastly, just to say, this is the effort of very many people. So we run annual workshops to train people in Oxford nanopore sequencing, again, with the help of Diplomics. And we actually take these opportunities to sequence small batches of these genomes that have made their way into the collection. And in that way, like I said earlier, we can train a lot of these folks. I just have to say a special thanks to Stephanie, who's the biobank manager, and Chelsea, along with Jo Marie Froilink. They did most of the heavy lifting in sequencing all of these genomes. And thank you for your attention.